Hey everyone, we're going to be looking at cell respiration, and there's a few things we need to know about before we can tackle that topic. So, cell respiration is how your cells make ATP. So we need to figure out what ATP is first and how it actually works, and then we'll talk about cell respiration, which is the making of ATP, maybe one of the top important processes that your cells do. So looking at ATP, here's our molecule here. You have this sheet in your Google Classroom. Look at its name, adenosine 5' triphosphate, or ATP for short. So this can take us back to some of the rules of naming these things. Remember, this molecule right here is a sugar. It's actually the sugar ribose. I'm going to label that on my sheet. Ribose is a 5-carbon sugar. At each of the little points, there's a carbon. There's one here, 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 and then up top here. There's not one at this point because an oxygen is there. So remember how these are named. This is called the 1' carbon, going from right to left. This is the 2' carbon, the 3' carbon, the 4' carbon, and then up top is our 5' carbon. And so that's why it's called adenosine 5' triphosphate, because off of the 5' carbon are these 3 phosphate groups. Remember your phosphate groups, the PO4, plus it's shown they have a little bit of a negative charge. They show up a few times in our biology. Remember cell membranes, the phospholipids also have a phosphate group with a negative charge. DNA will also have phosphate groups with a negative charge, and ATP has it as well. This molecule up front here is the same molecule that is actually in DNA. It's called adenine. It's called a nitrogenous base since there's so many nitrogens. So we have adenine, we have the simple sugar ribose, and then we have 3 phosphate groups. Now this is really extremely important here. This 3rd phosphate group there, that one is really on there unstable. As you keep adding phosphate groups, they get less and less stable of a molecule. So it turns out ATP, for as important as it is, it's really a very unstable molecule and really doesn't last very long, especially with this 3rd phosphate group on here. That's why we have to continually make it. We can't store this stuff. It would be nice if we could, but it's too unstable. It breaks down too easily, so we can't store it. We literally have to make ATP as we go. All right, so this last phosphate group is very unstable. Another important thing to remember, that with all of these different bonds here, with all these phosphate groups, there's potential energy or chemical energy. That energy is stored in the bonds. You can actually store chemical energy in those bonds, and remember, energy can be transferred. So when we break these bonds, which is a catabolic reaction or an exergonic reaction, when we break those bonds, energy is actually released and transferred to something else. So typically, for ATP to work, it has to break this last bond off of right here, off of this last phosphate group. That bond right there is broken. Highlighted in yellow, that's the important bond. That bond breaks off, and this phosphate group gets transferred to another molecule. When that happens, that's called phosphorylation. So I've got that right here. So how does ATP work? In this area, and then we have some examples off to the right. So first of all, in order for this to work, we need an enzyme. Remember, you have thousands of enzymes in your cells, and enzymes are proteins that do very, very, very specific jobs because their shape is specific, that specific 3D shape. Then they bond to their specific substrate, and in this case, a kinase bonds to ATP, and then another molecule to be able to transfer that phosphate to the molecule. So there's actually many different types of kinases, but they all have in common is that they bond to ATP and then another substrate, and they transfer the phosphate, this last one right here who's on there really unstably, they transfer that phosphate to that substrate. So that's the job of a kinase molecule. We've got the ACE ending, which is typical of an enzyme name, so it's easy to see that. So kinase molecules, so important because they grab onto ATP and another substrate. They help to take that phosphorus group off and transfer it to something else. So that whole process, once again, is called phosphorylation. Think of it as a zing of energy. Phosphorylated. The word even sounds like I'm getting energized. When I get phosphorylated, I get energized. So many of you will remember the sodium-potassium pump. It might be something like this, where there's your enzyme that runs the sodium-potassium pump, and it gets phosphorylated. Remember, that's one of the first steps, is to phosphorylate that enzyme so that it changes shape. That energy is transferred to that enzyme to change shape so that sodium can be pushed out of the cell and potassium can be pushed into the cell. If we did not phosphorylate that enzyme using ATP, that would not work. All right, let's see what number 2 says. The phosphorylated substrate molecule loses its P. So whenever phosphate is just by itself, we put a little I next to it. That means it's inorganic phosphate. So it's basically just a phosphate group, not attached to anything. It's all by itself. Inorganic phosphate. An enzyme that removes phosphate groups is known as a phosphatase. All right, well, that was another part of this job here, was to lose the P. So phosphatases can actually take P's away. So let's think of it this way. When a kinase enzyme grabs onto ATP, it grabs the P from the ATP and transfers it to another molecule. Think of it as like somebody's giving you candy. Think of the candy as the phosphate group. Everybody likes candy, right? Well, maybe not everybody, but in the cell world, being given a P or a phosphate group is like giving you candy. Yum. So that phosphate then helps you do work because the energy is transferred to you with that phosphate group. And then those enzymes that take away that phosphate group from you, it's like taking the candy away from you. First somebody gave it to you, now they're going to take it away? What? Oh yes, this happens all the time. So the phosphatase enzyme is what takes it away. When the substrate loses its phosphate group, it can use the energy released to do cellular work. The transfer of energy, one of the laws of thermodynamics. Energy isn't created or destroyed, just transferred continually, again and again and again. The PI, the inorganic phosphate, can then be recycled and added to ADP to create a new ATP. All right, that's the whole cycle that ATP goes through continually. We start with our ATP here. Then the P gets taken off. Yikes. There it is. The P is removed and this becomes an inorganic phosphate. But it was given to another substrate for a short while. It basically kind of made the substrate energized for a while so it could do work. And then, well, this ATP here is now ADP. It's adenosine diphosphate, di because it only has 2 phosphate groups, and then the inorganic phosphate by itself. This all needs to be recycled, so this is going to go back to the mitochondria so that we can put the P back on here. So it's like recycling. That's what the mitochondria does, is recycles ATP for your cells, continually putting the P back onto ADP to make ATP, so ATP can go back out and do work. Just like we see up here, to phosphorylate an enzyme pump possibly, or an enzyme channel or a protein channel or protein pump to help get things across cell membranes. It looks like this one is a muscle cell, and we have some actin and myosin here with the little cross bridges that need to be phosphorylated so that they can walk along this little protein filament. I think that's myosin, and then your muscles can move. That all takes ATP. I should put that right here, ATP. And so that's one example on top, the middle example, and then down here looks like we're going to put a P onto this monomer. Maybe this is 2 simple sugars and we want to make a disaccharide. So if we phosphorylate this, it'll energize it and then allow that bond to occur, because when this P gets dropped, energy is released so that this bond can be completed. Now we have a disaccharide. So what we're left with here after we use ATP then is ADP. As we showed down here, we'll have to recycle that. Your muscle cells use about 10 million per second each. And so we continually have to make this ATP as we go, because this last phosphate is so unstable. So moral of the story is it's all about the energy that's stored in these bonds. And if I give this phosphorylate, or give this phosphate to another molecule like I did here, probably uses a kinase molecule to do it. But if kinase can break off the P from ATP and give it to this substrate, then this substrate can use the energy right here in this bond to make another bond with whatever it's supposed to make a bond with to make a finished product. Alright, and then we have ADP. We're going to have to recycle that. That's going to go to the mitochondria, and that is cell respiration when we recycle this and put the P back on. Alright, so I've switched gears here a little bit. There's our ATP in color up in the corner, so now you should be able to recognize adenine, which is a nucleotide or a nitrogen base that also belongs in DNA and RNA as well. Ribose, which is a 5-carbon simple sugar. And the phosphate groups, 3 of them are attached to the 5' carbon of ribose. Ribose is the same simple sugar in RNA. The last phosphate group is that important one. It's on there really unstably, and we can give this phosphorus group to another substrate to energize it. That's called phosphorylation. Here's kind of a space-filling model of ATP down here, and a reminder that we make ATP in the mitochondria. That's called cell respiration when we do that. So we need energy for all these different things: building molecules, reproduction, movement, active transport, temperature regulation. That's going to come from ATP. All right. Remember, the work of life is done by coupling reactions together. If I have an exergonic reaction, I can use the energy that's given off by that to fuel an endergonic reaction. All right, let's follow this reaction down here. Looks like I want to digest or do a hydrolysis of maybe a disaccharide. And so, of course, that's going to take energy to do that. Over here, we're going to do a dehydration synthesis. Oh, excuse me, I got to go back here. Remember, the digestion is an exergonic reaction. That one actually releases energy. Whoops. I got to follow my pictures here. So that energy then is used here. I like this picture. That sends that energy down to this dehydration synthesis where I'm going to join these molecules and actually make bonds, but I need energy to put into it because this is the endergonic reaction. So if I do these reactions nearby each other, I can use the energy from one to fuel the other. That's called energy coupling or coupling of reactions. So to fuel this economy, I like that, we have to eat our organic macromolecules, carbohydrates, lipids, proteins, nucleic acids, and break them down and capture the released energy in a form that we can use. Oh, nope, we can't use a dollar bill in our cells, but we can use something else. We can use ATP. So basically, we're going to use the foods that we eat, carbohydrates, lipids, and proteins, and nucleic acids. We're going to use the foods we eat to make ATP. Again, it's even stating here, this is a short-term energy molecule. Not good for long-term storage. It's too unstable. So there's our ATP. ATP can come in many forms. When it's adenosine triphosphate, it means it has 3 phosphate groups. But it can also be adenosine monophosphate. Mono means 1. It can be adenosine diphosphate and have 2 phosphate groups. So it comes in many forms. It just depends how many phosphate groups are you going to have on there. Monophosphate, diphosphate, or our typical triphosphate when we are doing cell respiration. I like this. How efficient. Build it once, but you can use it many ways, and you can just modify it a little bit by adding different functional groups, like phosphate groups here. These are high energy bonds, like that picture. So each negative phosphate group is more difficult to add. There's a lot of potential energy stored in those bonds. It's like that, especially that last one, is spring-loaded on there. It just wants to pop off so easily, and when it does, it will release energy. So it's an excellent energy donor. Not good for storage, but an excellent quick energy donor because of that spring-loaded last phosphate group. Whoa, there it goes. All right, so now we took ATP and we just made ADP, adenosine diphosphate, plus the added phosphate group, the PI, and it released energy. Got some energy there, and it can fuel other reactions. When that phosphate group is given to another molecule, it's called phosphorylation. So one nice thing then, we can transfer that energy to other molecules. Who's the other molecules that want this energy? Well, proteins, of course. They're the workhorses of the body, but they need energy to do stuff. They need to be phosphorylated. Enzyme, all those different kinds. When you phosphorylate a molecule, you destabilize it. And it's done with that enzyme kinase. That's its job. So hopefully by stating these things a few times in this lecture, I hope this really sticks in your head for this upcoming chapter. So here's an example of phosphorylation. Here's 2 monomers of sugar. Remember how that's going to work? We're going to take an H off one of them and an OH off another, do a dehydration synthesis, and make water plus a disaccharide here. But it's never that simple. We need to destabilize the monomers first. We do that by phosphorylating them. Who's going to phosphorylate them? Well, of course, ATP will. So here's our monomer. We've got our ATP. We add that P, make a DP. That's going to go back to the mitochondria to get recycled. There's the phosphate group that's attached to this monomer now. This monomer has been phosphorylated, energized, destabilized. A kinase enzyme did that. Typically, again, we write that at the top of the arrow. So here's that kinase enzyme up here in purple. There it is, grabbing a hold of the substrate, the one monomer, and the other, it's going to phosphorylate and make us a disaccharide. So that monomer is going to have to drop that P, as we see here. It's going to have to drop it, so we get a PI, inorganic phosphate. That will also go back to the mitochondria to get recycled. Here's another example of phosphorylation. We're going to get into cell respiration, the making of ATP. There is a lot of chemical reactions that take place to make cellular respiration. We're not going to memorize those by any means, but we are going to look at the highlights of it. And one of the easiest ways to make ATP, and the fastest, most efficient ways to make ATP, is to use glucose. We can use fats and proteins as well, but using glucose is the fastest. Glucose is a 6-carbon molecule, and one of the first steps to actually make ATP is to break glucose in half. And in order to do that, in order to break it in half, we phosphorylate it first, adding a P on both ends. So in order to make ATP, we actually have to use some ATP to get it started. It's like you have to use a little money, spend a little money to make a little money. Actually, you make a lot of money in the end. Kind of like the stock market. You put a little money into it, and then you come out with even more. Well, if we put a little ATP into this process first, we'll come out with a lot of ATP in the end. So we phosphorylate our glucose, and it really makes it uncomfortable and really kind of destabilizes or energizes it. So it's much more likely that we can break bonds and make bonds and do all that stuff that we need to do. So here's an early example of phosphorylation in the process of cell respiration. Again, we cannot store ATP. It's too unstable. But to give you an idea, a working muscle recycles over 10 million ATPs per second. Yikes. Glucose is the fastest to do that, but we can use other macromolecules as well. So it's easy to say that cells definitely spend a lot of time making ATP in their mitochondria, and actually it's recycling ADP and Ps to make ATP. So now you know that a little more specifically, that the mitochondria is a recycler. Now look at the different folds in the mitochondria, folds in the membrane. When you put folds in there, you increase the surface area to have enzymes stuck on those membranes to make more ATP. So that's where the enzymes go. So here's another example of an enzyme. It's called ATP synthase. This is the enzyme responsible in the mitochondria for making ATP. It's embedded in these folds, on these folded membranes. Oops. Sorry about that. So ATP synthase, we've got the ACE ending on it. It's stuck in the cell membrane. This, if you look at it, actually has quite the shape. We can actually call this the world's smallest turbine. If you think like a water wheel, it actually spins. This is an enzyme stuck in this membrane that actually spins, and its job is to take ADP and P and make ATP. So how does it do it? Well, basically, we have to collect a lot of hydrogens. So it's not a water wheel, it's actually a little hydrogen wheel, hydrogen protons. And when these hydrogens flow down ATP synthase, it makes this enzyme spin, and it gives that enzyme energy to take the P and put it back onto ADP to make ATP. Right? So this is the enzyme that recycles ATP. Pretty awesome. And these are in the mitochondrial membranes. So the idea here is that we have to gather hydrogens in this spot and then make those hydrogens flow down. It's easy because it has an affinity for hydrogen. It's very permeable to hydrogen. So now the question is, how is this hydrogen gradient formed? Where do we get these hydrogens from to make this precious ATP? Well, the answer is cell respiration. We have to break food down and gather the hydrogens from the food that we eat, all to take it to the mitochondria, take these hydrogens here so they can flow down our ATP synthase, make it spin like a water wheel or a turbine, and put the P back on to ADP to make ATP. Alright, so that's ATP, and phosphorylation and energy and all that, all to get us into cell respiration.